Hello, everyone. Good morning and good evening. Welcome to the Asia Molecular Diagnostic Laboratory, Pillar Biosciences joint webinar on precision oncology in companion diagnostics. I'm William Chen, Head of R&D at AMDL, and today's Master of Ceremony. AMDL is the joint venture between Pillar Biosciences and China Biotech Services Holdings Limited to introduce Pillar Biosciences cancer next-generation sequencing panels and services to Hong Kong and the wider Asia region, including the first FDA-approved ONCO Review companion diagnostic test for non-small cell lung cancer and colon cancer, which is done locally in our laboratory at Hong Kong Science and Technology Park. Pillar Biosciences' proprietary technology, SLIM-M, allows the test to be completed in shorter time, at lower cost, and with excellent accuracy and sensitivities. In today's webinar, our speakers, including experts of Pillar Biosciences from the States and China, will tell you in detail about the technology and how it can help you and your patients on therapeutic options. Before we move on to the next section, may I remind our attendees, if you have any questions during the talks, please click the Q&A tab at the bottom of your screen and type your question there. We will invite our speakers to answer the questions in the last part of today's program. Thank you. Without further ado, may I introduce our first speaker, Mr. Wing Tang, Executive President of China Biotech Services Holdings Limited and General Manager at AMDL. Mr. Tang is an experienced business developer and innovator and has served in several international pharmaceutical companies and clinical trial companies. He is also serving at several government institutions to develop and promote the biomedical industry in the region. May I now invite Mr. Tang to open the webinar and welcome you all. Mr. Tang, please.
Good morning and good evening to everyone, because our guest speakers is from the United States, so they are in a different time zone. So it is my great pleasure and honor to welcome all of you. Thank you very much for joining our webinars today, the first FDA-approved lung and colon cancer companion diagnostic test in Hong Kong, with FDA-approved prescriptions of targeted therapies. My name is Wayne. I'm the Executive President of CBSH, China Biotech Services Holding. CBSH is a listed company in Hong Kong, and our investments are focusing in medical and healthcare sectors. For example, in our business model and investment sections, we offer the dietary cancer genetic tests and early screening and cancer risk genetic tests. Besides that, we own one of the well-known and oldest central lab in Hong Kong, PHC, Medical Diagnostics Center, 標準病理檢驗所. This year, our company has reached the 54th years old. We also have several clinics, VK Medical Examination Center, offering health checkup, vaccinations, COVID-19 antibody testing, as well as other health management and customized services to corporate clients, insurance companies, pharmaceutical companies, and so on. Not to mention, VK has over 28 years old too. During the pandemic seasons, CBSH and BGI joint venture and established Sunrise Diagnostic Center for precision diagnosis of COVID-19. It has a capacity of testing more than 130,000 samples per day. And we are one of the largest contractors which the Hong Kong government has been appointed to offer Hong Kong nationwide COVID-19 screening. And one of the popular projects that well known to all of us in Hong Kong is a 火眼 laboratory, 香港火眼實驗室. Of course, we are well aware of the increase and the demand in cancer treatment globally. We therefore further invested 2 more companies to offer cancer treatments with cutting-edge technology. They are CAR-T treatment and BNCT, boron neutron capture therapy. Our CD20 CAR-T OX40 clinical trial is the first CD20 CAR-T approved for clinical trial in China, and one of the first CAR-T CAR-T products with CD20 with IND approvals in the world as well. So far, our clinical performance has been very well, and the result is, and the efficacy is very good too. Soon next year, we will share some more of our data. In addition, BNCT treatment is the only approved BNCT treatment device in the world. It can selectively and accurately kill the tumor cells at the cellular scale. It can increase the survival rate from 4% to 79%, especially on malignant glioma patients. This BNCT treatment has been approved by the Japanese PMDA, covered by their national health insurance today. CBSH is also currently developing recombinant subunit vaccine for the SARS-CoV-2. This technology was originated from China CDC by the name of BizaVac. We also offer value-added services to our healthcare industry by offering drug registrations, distributions, importations, warehousing, logistics services for pharmaceutical drugs and medical device. As you can see, CBSH is very focused and specialized in precision diagnostic sector. According to statistics, the global potential market forecast for cancer companion diagnostic tests will raise from $5.5 billion to $9.9 billion from 2022 to 2025, with the CAGR values of 12.6%. How about APAC? Asia Pacific forecasts the raise from $0.79 billion to $2.37 billion, with the CAGR values of 24.7%. APAC, Asia Pacific, projected to be the fastest growing region, 2 times the global growth predictions in the coming 5 years. Today, I sincerely like to share with you, it is our honor to have the JV partnership with Pillar Biosciences. On January 2019, CBSH entered a partnership agreement with Pillar Biosciences to deliver precision diagnostic tests to the Asia Pacific market. We therefore set up AMDL in the Hong Kong Science Park, a NGS-based molecular lab offering CDx services and distributing products in China, Hong Kong, as well as Asia Pacific. AMDL is also a CAP accredited laboratory too. Pillar Sciences is one of the 3 CDx approved by the US FDA today. Pillar Sciences received the FDA pre-market approval for its ONCO review DX lung and colon cancer assay in 2021 with 2 patent technology, SlimM for targeting sequencing and PIVAT for data analysis. Today, it is the first FDA-approved CDx to be launched in Hong Kong without the necessity to send the sample to overseas for tests. As such, we could offer you a much faster turnaround time, definitely less than 1 week, simplified workflow, and of course, a much lower cost. I guess this is what all of you are interested today. Now, I'm excited to introduce our guest speaker today, Dr. Gang Song. Dr. Song received his PhD from Shanghai Medical College of Fudan University, China, and completed his postdoc in Timothy Springer Lab at the Harvard Medical School. Dr. Song is the founder and CEO of the Pillar Biosciences, who co-invented Pillar Biosciences SlimM technology. He subsequently founded Fit Immune Therapeutics, a cellular immunotherapy company. Prior to that, Dr. Song was a technical lead at the Integrated Genetic Lab Corp, previously known as the Gemzyme Genetics, and he was also the project lead at iQuome before its acquisition by Roche Molecular Diagnostics. Dr. Song is definitely the professional in precision and companion diagnostic tests with in-depth of experience. Let us welcome Dr. Song and listen to his story of Pillar Biosciences. Dr. Song, please.
Good morning and good evening. Today, I'm going to introduce you Pillar's next generation testing sequence, NGS testing solution. At Pillar, we aim at advancing decision medicine through empowering decision-making process across the entire healthcare journey for everyone, everywhere. That's our belief. Pillar was founded in 2014. By now, Pillar has global presence. Our headquarters is in Boston. We also have operations in Shanghai, China. As Mr. Tan introduced, with CBSH, we established a joint venture in Hong Kong to serve the Asia Pacific market. Pillar is a product-based company, very different from most of other oncology NGS companies, which are service-based. We built products based on our proprietary best-in-class technologies. The whole workflow is fully automatable to enable NGS testing at any lab. Then all the hospital labs, cancer labs in cancer centers, third-party reference labs, and in pharma, they can eventually, if they adopt all our Pillar's products, they can do NGS testing locally, closer to the patient. As Ms. Tan introduced, last year, Pillar received first ever FDA PMA approval for multi-cancer NGS CDx. This same product has also obtained CE mark in EU, and we are also anticipating NMPA approval later this year. At Pillar, our purpose is to make cancer and other complex disease manageable through actionable genetic insights. Our mission has always been to enlighten and simplify the healthcare journey with our leading innovative technology to empower better detection, treatment, and prevention of complex disease for everyone, everywhere. Here, I'm sure everyone has been familiar with precision medicine. Precision medicine based on everyone's biomarkers, either genomics biomarkers or protein biomarkers. The oncologist can treat a patient in a more personalized way, more precisely. However, the precision medicine has encountered many challenges. In particular, in clinical NGS testing, there are many challenges. The first one is high failure rate reported. Very recently, in a very extreme example, AstraZeneca reported their clinical trial called PROfound trial for their olaparib, a PARP inhibitor drug. They used the Foundation's test to test the all the samples from this clinical trial. However, only up to 58% of samples they actually received reportable results. This means as high as 40% of clinical samples, there was no reportable results, just because the Foundation's panel requires very, very high quality samples and with high gene input. But very often, as you know, clinicians, oncologists, and lab, they encounter clinical samples often with very low quality and low quantity, like a fine needle aspirate. So at Pillar, in the past 7 years, we have been trying to overcome these challenges by innovation. So we developed our technology platform from scratch By now, we built a portfolio of products that can decrease the testing failure rate by over 95%. This has been validated by many customers and collaborators across the world. Then the test is a lot more cost-effective with faster turnaround time and can be localized with highest lab efficiency. Here are just some examples. We're trying to break the bottlenecks in precision NGS tests. We built robust IVD and IVD grade products to serve the needs of different channels. The first is hospitals. At hospitals, they typically don't have expertise to provide very sophisticated NGS tests. Typically, they just want packages of IVD products. Then they can serve their patients. So this is why we, after we founded Pillar, from early start, we wanted to build IVD products in NGS. The first one is ONCO Reveal CDx tests. We also built a portfolio of IVD grade IO products. The second channel is farmers. With farmer, including AstraZeneca, we have built custom panel for HR deficient metastatic breast and relapsed ovarian cancers to support phase 2 trial to see whether we can identify more patients that could respond to olaparib. Among cancer centers, which Helen Fernandez at Columbia University Medical Center will introduce you their LDT version of Pillar solid tumor panel. Also, we serve third-party labs, including lab called KingMed, BGI. Then they can use our either customized product or standard products. Then they validate these products in their CLIA labs or reference labs to serve their patients. So what's our technology platform? We have built 2 major platforms. In the middle is a chemistry. That's a SlimAmp chemistry. This is a technology that can allow parallel amplification of hundreds of or thousands of amplicons in a single tube reaction. In this way, the NGS enrichment is simplified. To enable automatic design of SlimAmp libraries panels, we also built this AI-driven versatile primer design platform. Any contents we can use versatile to design the panel in just a few minutes to a few hours. This allows ultra-high plex amplification of any NGS panels. So the NGS panel from this SlimAmp technology is just a single-day workflow. It's fully automatable at any liquid handlers with very low cost and very low assay failure rate. We have obtained global patent rights for these technologies. Downstream, we also built our Pivot data analysis platform to enable data analysis from raw data to actionable reports. This is just an example of data from our PMA study. In PMA study, the number one criteria that FDA looks at is actually reproducibility, not sensitivity or specificity. To the FDA, reproducibility is most important because this is going to be a kit. Then every lab in any hospitals, they should be able to repeat with the same results. Here, just show our FDA PMA panel was tested at Columbia University Medical Center, at Dadamas Hitchcock Medical Center. Monica MD is part of ICON. It's a CRO company. In all three labs, they tested the same set of samples, and the average library prep time is was only about 6.5 hours. With automation, this 6.5 hours can be further reduced. The average sample to pipette results, the time was 47 hours, just within 2 days. Compared to other decentralized solution like Archer Invitae's solution, and also other centralized lab solution, including Natera, Signatera, Guardant 350, Foundation One, our Pillar tests has the shortest workflow, most robust results. Now I will introduce you this PMA product. As I mentioned from the very beginning, we want to make sure our products can be regulatory compliant. Starting 2019, in collaboration with Helen Fernandez at Columbia University, they made a customized solid tumor panel called ONCO Reveal Solid Tumor Panel. They validated in their lab and submitted to New York State for approval. New York State approval is the highest standard in LDT industry. Then in 2020, our ONCO Reveal DX lung and colon cancer assay was CE marked. In 2021, this was approved by the FDA as PMA. Now we are trying to expand our claims and indications from just lung cancer and the colon cancer CDx to pan cancer. We expect to submit this supplement to the FDA next month. At the same time, we are working on other products to be submitted to the FDA in liquid biopsy. These are the claims, so-called Table 1 claims. So FDA actually approves tests with different tables. Table 1 is CDx claim. That's the highest standard. In our product, these are claims for specific drugs. The highlight here is for the EGFR, we actually obtained a class claim. So any approved EGFR TKI inhibitors by FDA or any EGFR TKI that will be approved by the FDA in the future, these drugs can all use our PMA product as a CDx. So this is so-called class approval. This is clinical concordance data. During the clinical study, we compared our tests with FDA-approved tests here for EGFR, that's Roche Cobas EGFR mutation test. For KRAS, that's QIAGEN's FDA-approved screen KRAS test. Because any comparator must be FDA approved. There are not really many out there. So as you can see, the PPA and MPA are very high. So any non-concordance data, FDA actually does not recommend to address them. However, we actually know our results are real. So here are the detailed data. As you can see, our test compared to EGFR, Cobas, the concordance rate is very high, and same is true for the KRAS test. The EGFR Roche Cobas, because their cutoff level is 5%, this is why they missed a few calls with VAF between 1.9 to 4.9%. In the case of seroscreen test for KRAS, because QIAGEN's test was not designed to detect complex mutations like here, KRAS C.3435 dial-in TT mutation. The seroscreen actually couldn't capture this. This is why our results are discordant. Actually, no, because this is a sequencing test, we can capture those. We also wanted to compare our test with foundation tests, but after we contact them, they were not interested in such comparison for apparent reasons. So here is the DNA input study. We tested DNA input from as low as 5 nanogram to 160 nanogram. Our final recommendation of DNA input is 10 to 80 nanogram. However, as you can see, at 5 nanogram DNA input or 160 nanogram DNA input, the PPA, MPA are still 100%. Again, this is detailed data about reproducibility. We asked the 3 sites, and each site, there are 2 operators. They performed 3 runs on non-consecutive days. Then 1 sequencing instrument and 2 reagent slots were used at each site with 10 clinical samples. As you can see, for all the mutation sites, the PPA and MPA were 100%. Then what about tumor content? In clinical settings, tumor contents can often be very low. Foundation tests, the tumor content cutoff is as high as 20%. But in many cases, as shown in AstraZeneca study, their clinical trial, many samples failed this cutoff. In our PMA study, we tested as low as 10% or even below 10%. As you can see, we could get reportable data from 10% and above. For tumor contents even lower than 10%, as long as there is still tumor content, 96% actually were successfully called out with reportable variants. This is an accuracy study. We compared our test with thermoofficial Oncomine focus assay, which was validated as a LDT at Molecular MD. At Molecular MD, they tested 208 samples. 84 of them are CRC, 124 are non-small cell lung cancer. They tested this same set of samples using our assay and Oncomine focus assay. And the concordance is pretty high, as high as 98%. Again, because of the assay cutoff of Oncomine focus assay was 4%. So with those positive results with less than 4% VAF, Oncomine focus assay couldn't capture it. However, our assay caught these mutations. So now we are working on this so-called PMA supplement. In this supplement, we are going to expand our claims from CDx indications in EGFR and KRAS to other gene targets with evidence of clinical significance. There are 22 genes in this panel. We expect to receive this approval later this year. Then we can offer pan-cancer PMA product global-wide. So I have introduced you the PMA product. However, as everyone knows, it takes really long time. In this case, like over 3 years to just get one PMA product. So patients cannot wait. This is why we also have been working on so-called, we call IVD grade IO product line. We will move some of them all the way to the PMA process. Then many of them, we will keep them as IVD grade. IVD grade means we developed these products using the same QMS, and we are going to test all these panels with large number of clinical samples and do analytical studies. The only thing we cannot do for every panel is a clinical study. Only those PMA products we will do clinical study. So this slide shows you our product line. Already talked about tissue biopsy panel. The next is ONCO Reveal HRD plus methylation panel, which will be used for public inhibitors like AstraZeneca's olaparib. We already launched IVD grade IO product of the HRD panel. We're adding methylation piece. We expect to launch this combo by the end of this year as IO launch. In liquid biopsy, we are going to launch ONCO Reveal core panel very soon, and ONCO Reveal enhanced panel early next year, and this MRD detect panel, which is a tumor-informed MRD monitoring panel, later this year. So in liquid biopsy, we offer a different set of panels, ranging from the small panels, like specifically focusing on lung cancer, liquid biopsy panel, which consists of 7 genes, like EGFR, KRAS, those genes, for specific drug. Then in the middle, this is our core panel. which includes targets in 106 total genes. As comparison, Guardant CDx has 76 genes with very high sensitivity at 30 nanogram of DNA input, 0.1% VF. Then this MRD, a tumor-informed panel, we can include as high as 45 patient-specific SNPs. And more important, because of our the power of our SLIM technology, we will be able to develop an off-the-shelf primer bank for tumor-informed liquid biopsy monitoring. As you may know, Natera Signatera, because their technology can only multiplex up to 16 amplicons, they won't be able to include 45 patient-specific SNPs and won't be able to make this off-the-shelf primer bank. Finally, these are our standard oncology-focused IVD and IVD-grade products. The yellow highlighted are panels with lots more customers. So they all want to use, adopt, test, and adopt our panels. So I won't go into details. If you have any questions in our Q&A, we at Pillar, we have our team like Shiping, our global director of product management, and our Zhenggu team. And with our AMDL colleagues, we can answer any questions you may have. Okay, I think that's it from me.
Thank you, Dr. Song. I think we are all impressed by the excellent performance of the ONCO revealed DX lung and colon cancer assay, and the wide variety of products available in the pipeline that surely can benefit clinicians and patients in Hong Kong and Asia. May I now invite our next speaker, Dr. Helen Fernandez, Professor at the Department of Pathology and Cell Biology, Columbia University Medical Center. Dr. Fernandez is an expert in molecular cancer testing and has validated Pillar Biosciences products in her laboratory. Today, she will share with us the clinical data of the validation of ONCO Reveal solid tumor panel for the Department of Health of New York State. Due to packed schedule, Dr. Fernandez cannot attend the webinar in person, but she kindly provides us with the following video recording of her talks.
Hello, everyone. My name is Helen Fernandez, and I am a professor of pathology at Columbia University Medical Center in New York. As part of my responsibilities, I oversee the validation of laboratory-developed tests, or LDTs, that are submitted to New York State for approval. Today, I'm going to present my experience on the validation of a 50-gene targeted next-generation sequencing panel, or NGS panel, for the detection of mutations or variants in solid tumors. The NGS panel that I validated uses SLIM proprietary technology developed by Pillar Biosciences. If you are testing a clinical specimen in New York, or if you're testing patients who live in New York, the LDT must be approved by New York State before it can be used for patient care. This slide shows you the different genes in the panel that was sent to New York State using SLIM technology from Pillar Biosciences. Again, the one on the left was the version 1 with 47 genes. The one on the right has 48 genes. We added one more gene. If you look at it, the panel on the right, the genes in red and green represent the genes that were fully validated. And if you count them, there are 17 in total, which means we had 10 specimens per gene that is highlighted in red or green. For 17 genes, we used 170 specimens for the version 2 validation that was submitted to New York State. This panel mostly is a hotspot panel that targets regions of the gene that are important from a diagnostic, prognostic, or therapeutic standpoint. But some genes are covered in full, such as TP53, STK11, and PTEN. This panel detects mutated genes in the following cancers that are listed on this slide. So for accuracy, in the first version I'm going to talk about, we identified 136 variants or mutation that were harbored in 108 samples. In addition to that, we had normal or wild-type specimen, and we also had DNA from well-characterized cell lines, such as listed here, NA12878 or genome in a bottle. These are really well-characterized, and New York State wants you to use these samples in your validation as well. So as you see in the 2x2 contingency table over here, we obtained a specificity and a sensitivity of 100% in this validation that we did for the solid tumor panel, which is excellent. This slide is a graphical representation of the performance of this well-characterized cell line NA12878. Each bar represents the variant allelic fraction from 6 different runs. These, on the x-axis is the different variants that we interrogated, and on the y-axis is the variant allelic fraction. This is a characterized cell line that is either homozygous or heterozygous, which means it has 100% allelic fraction or 50% allelic fraction. What I want to bring to your attention is these bars also have error bars in them. And as you see, these error bars are really very tight in most of these cases, which tells you that the performance of the assay was very good. So to summarize very briefly the accuracy that we sent to New York State, there was 100% concordance between the 136 variants in version 1. The genes listed there on the slide are the genes that were validated in full, which means we tested 10 specimens per gene when it indicates if it was SNVs or indels. We tested many different kinds of specimens listed here. The results we obtained were from specimens when the DNA input was really low, 1 to 3 nanograms. The variants are detected in DNA obtained from samples with as low as 10% tumor in the specimen. And the analytical accuracy of patients is really good even when the DNA input is very low, 2 nanograms per reaction. The average coverage of specimens in our particular validation was about 10,000. This actually varies depending on the number of samples are assayed in any one particular run on one particular flow cell. So the next thing we see is the analytical sensitivity. And you have to establish this for each variant in the assay. I've shown you on this slide just representative graphs from 2 different variants on the left side and 2 different variants on the right side. So the data on the left shows you that alterations were identified in tumors with allelic frequency as low as 2.5%. So even when it is 2.5% here, you can detect this particular mutation. The 2 mutations that you see on the slide, this is a deletion in EGFR, exon 19 deletion, and this is a single nucleotide variant in the gene PI3 kinase or PIK3CA. On the right side, in these back graphs, what you see is that these alterations are identified even when the DNA input is very low, or 2.5 nanograms. So it's very sensitive. The next items to address are precision and reproducibility. Precision is also known as repeatability. So to do this, we took 4 or 5 different specimens and mixed them together to form a single mixture which had these 5 variants that are listed in the slide. This was run in triplicate on one day, run in triplicate on the second day, and run in triplicate on the third day. Just want to bring to your attention very briefly that when you compare the variant allelic fraction obtained in a replicate run on day 1 in all 3 replicates, as well as the replicates run on day 2 and 3, the standard deviation is excellent. The precision of this assay is very good. On this slide, similarly, the reproducibility of the assay is very good. To do reproducibility, you have to run at least 4 or 5 separate runs using different barcodes to make sure that you know that these are different patient specimens. Now, these are different specimens done in different runs. They should be run on 3 different days and run by at least 2 different technologists. This is to show the variability on days and variability between technologists. And if you go into the slide in detail, it is pretty good. Some additional notes to talk about over here. When selecting specimens for validation, one needs to actually select the tough ones. Which are the tough ones? They should include a representation of specimens that have high GC-rich areas, the ones that have homopolymeric regions that are hard to sequence, or if multiplexing samples with different barcodes, you should verify that there is no crosstalk between the barcodes. There is no cross-reactivity. And keeping in mind, when you're doing a validation, you should not change the version of the software. It should always be the same version. The next few slides deal with what we call quality metrics, which is very, very important for monitoring the performance characteristics of the assay. So I'm going to go very briefly over these. These are pretty self-explanatory. So this slide shows you the coverage distribution of the assay that was submitted to New York State. At 300x, we get excellent coverage. At 500x, we get 99% of the coverage of the samples that show you good coverage, on 99.2%. At 1000x, 96.3% of the specimens have at least 1000x coverage. So we submitted to New York State that we will accept anything that has at least 500x coverage, because 99% of the samples have been there. Monitoring the variant allelic fractions in multiple variants is important, and that is the function of the positive control. What you see in this slide is multiple variants that are present in the positive control that was used in every single run. This positive control contains 22 different hotspot variants that are at pretty low allelic fractions of 4% or 8 to 10%, between 4 and 10%. This is a Levi-Jennings graph that shows you the average of the average variant allelic fraction of each variant in this positive control. And it also shows you the standard deviation, a plus standard deviation of 3 or a minus standard deviation of 3. This positive control should be used in every run to assess the performance. And one should use this to every month to go over the data to assess the ongoing performance of your assay. It's a very good representation of the ongoing performance of the assay. And finally, this is a box and whiskers plot to show you the coverage at every single amplicon in this 50-gene panel. So each blue line here represents one amplicon. And as you can see, as would be expected, some amplicons are covered really well, some are not covered really well. But they're covered, all of them go at least above 1000x coverage. The ones that are not covered very well, as we talked about earlier, are probably because they are GC rich, or they have some homopolymeric regions, or just because the kinetics of the reaction is different for those amplicons, the ones on the left towards the left versus the ones towards the right. What we did in our particular validation is to compare 2 different pipelines, bioinformatic pipelines. We compared a pipeline called NextGene that was used to interpret, to look at the variants in our FASTQ files. We also ran these FASTQ files through the PyVet pipeline from Pillar Biosciences. And as you can see, there was great concordance between the 2 pipelines. So in summary here, the solid tumor panel uses next generation sequencing to uses Pillar's SlimAmp technology to successfully validate a 50-gene hotspot assay in our lab. This was submitted to New York State and it was approved by New York State. We included single nucleotide variants, indels, and splice site variants in the MET gene that were present in formalin-fixed paraffin-embedded tumor for validation. We used very low DNA input and low tumor percentage for the validation. In my final slide, I'm going to show you some data that represents the excellent performance of the solid tumor panel. Over the 2 years that we tested almost 2,000 specimens and successfully sequenced about 95% of them. Only 1.5% of samples failed because the DNA input was low or the quality of the DNA was not really good. So this is excellent performance when only 1.5% fails of that. The other failures were due to order error, or there was insufficient tumor, which was not estimated in the right way. Of the almost 2,000 samples that we did, 35% of them belong to lung, and about 30% of them were colorectal cancer. And I put together this pie chart just to show you the different mutations that we identified in our lung cases. And as you can see over here, it very much fits the picture that one sees in the literature, where the greatest number of mutations one sees is KRAS and EGFR, followed by some of the other ones that you see here. So having said that, I have presented to you how we validated our 50-gene panel at Columbia University, how we submitted it to New York, what we submitted to New York State for approval, and how we get approval for this particular assay. I also showed you how it performs and how we analyze patient data in here. I hope this was a good presentation. If you have any questions, please do not hesitate to contact me and I'd be happy to answer them. Thank you.
Thank you, Dr. Fernandez. As demonstrated by her data, the OncoView solid tumor panel is a very robust test for multiple cancer types to help clinicians and patients to make therapeutic decisions. This solid tumor panel is available at AMDL for clinicians in Hong Kong and the wider region. Now, may I invite our last speaker, Dr. Jacqueline Tam, Deputy General Manager of AMDL. Dr. Tam is an experienced scientist with more than 8 years of experience in next-generation sequencing, cell-free DNA, oncology, and prenatal testing. And she will introduce AMDL and our service to you. Dr. Tam, please.
So I'm the Deputy General Manager of AMDL. So I would like to introduce AMDL and our service provider. AMDL is a subsidiary of China Biotech Services Holding Limited. We are the JV partnership of Pillar Biosciences in Asia, and we are the sole distributor of Pillar Biosciences product. With our extensive company infrastructure, we would like to bring the world-leading NGS-based clinical testing service and product for better healthcare management to the public in Hong Kong, Great Bay Area, and Southeast Asia. So over the past year, AMDL and Pillar Biosciences demonstrate significant milestone and achievement. In 2017, Pillar Biosciences has been granted for patent in US and China for its SlimM technology. So in 2019, Pillar Biosciences entered a strategic partnership with CBSH to deliver precision diagnosis to the market in Asia. So in 2021, Pillar Biosciences received the FDA pre-market approval for its ONCO DX lung and colon cancer assay for KRAS and EGFR target therapy in CRC and NSCLC respectively. So in 2022, AMDL has passed the CAP proficiency test with a perfect score. This demonstrates our capability and quality for our NGS testing in cancer diagnosis. So just within a year, in the coming July, we are going to launch the first FDA-approved lung and colon cancer DX test from Pillar Biosciences in our Hong Kong laboratory. We aim to provide a locally conducted FDA-approved companion diagnostic test to the clinician in Hong Kong and Asia. So now I will show you our central laboratory located at Hong Kong Science Park. So here is our laboratory setup. We have equipped with the Illumina MySeq DX sequencer, which is the first MySeq DX in Hong Kong with the FDA regulated and CE-IVD marked for in vitro diagnostic testing. So there are 3 roles of AMDL. As a clinical diagnostic lab, we offer FDA-regulated and CE-IVD marked platform for cancer diagnostic tests, and we also offer cancer genetic tests to the clinicians. The second role is that as a distributor of Pillar Biosciences, we provide a variety of target cancer panel and bioinformatic analysis services. As a sequencing service provider, we offer fast and high-quality sequencing service to research institutions and laboratories. This slide shows the tests we offer. The first FDA-approved NGS-based DX lung and colon cancer test. In the meantime, we also provide NGS-based cancer genetic tests, which include lung and colon cancer panel, solid tumor panel, myeloid panel, pan-cancer 5080 gene panel, and a pan-cancer HRD panel. This slide shows the assay detail with gene, variant, and target therapy of our FDA-approved DX assay. Here is our test workflow. When there are tissue block or slide ready at the hospital or clinic, the doctor can simply fill up our sample request form and notice us by email or fax. Our logistic courier will come to the designated place within 2 hours for specimen pack up. So after that, DNA is extracted from the collect specimen, followed by a series of steps, including the library preparation, target amplicon sequencing, and NGS sequencing, performed by our professional team. In a couple of days, the genetic data is done. So with our guaranteed test service TAT, the clinical diagnostic report will be sent to the doctors for their medical decision on appropriate target therapies. So what information is provided in our clinical report? The summaries of findings. The number of variants with clinical significance, the number of variants with target therapies, and the number of variants with clinical trials. We have also identified the gene of variant with actionability and pathogenicity, followed by their corresponding approved therapeutic options. Besides, there are additional information of clinical trials for the variant and gene detected. Our clinical report utilized the international golden standard database such as COSMIC, TCGA, ClinRAF, and HGMD. We have update frequency in different knowledge base of clinical trials, drugs label, scientific literature, and international clinical guidelines. Our clinical report database is widely used worldwide, such as the government body, NIH, the global leading molecular diagnostic company, Nextera, Myriad Genetics, LabCorp. And in Asia, the reference sites include the National Cancer Center in Japan, Hong Kong Adventist Hospital, Prince of Wales Hospital, and the Chinese University of Hong Kong. So more reference sites will be established in near future. There are several advantages of our cancer diagnostic and genetic tests, which include the simplified and standalone workflow from sample collection to reporting with fast TAT. So we have satisfactory assay performance with high accuracy and sensitivities. We are the first FDA-approved DX test implement and locally conduct in Hong Kong. We have well-trained professionals governed by the MLT regulations on laboratory practice compiled to CAP standard. And we have very strong technical and bioinformatics support. So the FDA-approved diagnostics test is very important, which is meant that the assay is analytically and clinically validated for its intended use. AMDL DX test guide prescription of both CRC and NSCLC target therapy for accurate and precise treatment decisions. So there are 3 FDA-approved NGS-based CDx tests in cancer diagnosis. That is one, the Pillar Biosciences, Garden Physics Zero, and Foundation Medicine. Our advantage over others is the first FDA companion diagnostic test conducted locally in Hong Kong without sending test service to US. This greatly shortens the turnaround time with a reasonable diagnostic test for clinical report delivery to the clinicians for efficient clinical and therapeutic treatment decision. We provide different local and overseas test service to address your need. With our promise, AMDL provide a comprehensive and concise report for your clinical decision. So AMDL not only just provide the diagnostic test service. As a distributor of Pillar Biosciences, we do offer a variety of target cancer panel for laboratory research and development. So here is the list of target cancer panel with our top 5 products highlighted as yellow, such as the multi-cancer panel, myeloid cancer panel, and the BRCA1/2 panel. For those who are interested, please feel free to contact us by email, and our technical team will address your inquiry.
Thank you, Dr. Tam. As you can see from her presentation, AMDL laboratory at Hong Kong Science and Technology Park is the state-of-the-art laboratory that is ready to serve you. Here comes our last section, the Q&A section. I noticed that it is the expected end time of this webinar. We will continue the Q&A, but in case you have a packed schedule and would like to leave, but you have a question, please leave your question or send us your question through this email on the screen so that our technical staff or Pillar Bioscience experts can reply to your question through email later. I already see we have several questions in the Q&A box. So the first question is by Raymond Tong. He's asking whether AMDL is only selling surface, providing surface in Hong Kong, whether we would also sell products like Pillar Biosciences product to laboratory to do their own tests. So I think Dr. Tam has already briefly covered this part, but maybe you could further elaborate it a bit to Raymond.
So thank you, Raymond, for your question. So as I have mentioned before, as a distributor of Pillar Biosciences, we do offer a variety of target cancer panel for laboratory research and development. So I will show you the slide of our target panel available, including the number of genes, the sample requirement, and our sensitivity and specificity. So we offer an end-to-end comprehensive NGS solution from primer design, library preparation of simple and single tip workflow, sequencing to bioinformatics analysis and support. So please let us know your project plan and scope, and our professional team will address your need and requirement. Yeah, thank you for your question.
There's more question in the Q&A. I think it's along the line whether laboratory scientists or clinicians can can buy the panels. I think Dr. Tam makes it very clear, and there's a lot of panels available to purchase through AMDL, and also we could the p-vector bioinformatics analysis is available to you. So it will be very simple. Just you input the raw data, and then you would get the clinical actionable report. I have seen there's a question in the chat box by Clara Ip. So Clara is asking, the CDx product is already gained CE mark, yes, in 2020. Are they in the process of transitioning to the new regulation? So I think perhaps Dr. Song or Pillar Biosciences expert can explain a bit about the registration process. Dr. Song?
Yeah, sure, I can cover this. So yeah, thanks for the question. In Europe, they're changing the regulatory path from IVD-D to IVD-R, IVD regulation. We are in the process of making our panels to be IVD-R compliant. So for any new products that will be launched as IVD in Europe, we will go through the IVD-R.
Thank you, Dr. Song. I think yes, that's thank you, Kara, as well for your questions. Let me see. I think there's one more question in the Q&A box, which is the question is actually asking what is the advantages of Pillar Biosciences CDx lung and colon test over competitors on the market? I saw Shipeng, you are typing the answer. Perhaps, would you want to speak up and directly respond to it? You can unmute yourself.
Yes, I can. So a couple of quick things, right? As Gang has already introduced the panel to you guys. So I think that the major advantage is the low fail rate, right? We only need a low amount of input and then almost guaranteed to give you a results within a reasonable turnaround time. That is the major advantage for us to compare to our competitors. And Jacqueline also shows you that if this test is being performed in Hong Kong, the turnaround time is, obviously advantageous to our competitors. We have our FDA-approved SSED, and you can compare that with our competitors' sensitivity and limit of detection, which Pillar also shows advantage on that end. And also our test is more cost effective than those in other competitors. So I think that's the major advantage.
Thank you, Shipeng. I think Wayne, you're raising your hand. Would you?
I seems that I have troubles using this. I'm not so familiar because we have a guest from Japan that just sent me a questions. They're asking about the connections with the Japanese NCC. Shipeng, are you able to reply to these questions?
No, I actually was not.
There is a slide that is it from Jacqueline that there's a relationship with the in Japan with the National Cancer Center, the NCC.
That slide is not a relationship with NCC. So it just the NCC used the clinical database, which is the same as us. It is not a connection. Yeah, I just want to clarify just at that point.
Okay, and then also I'd like to read illiterate to Dr. Raymond Tong questions. He was asking about the whether it is only selling in Hong Kong. To answer to Dr. Tong is that we started with Hong Kong, but we are not limited to Hong Kong. We have already started marketing into the Asia Pacific region, which is the responsibility of our group to distribute all the services and products. Yes, we also sell the products. They can do the test at their own lab, or they can also send the product, or I mean the sample, to our Hong Kong lab, and we will send them the reply within a week. So this is what we are doing too. Okay, thank you, Williams. I return back to you. Thank you.
I just want to add one more comment to Dr. Teruyoshi Okuda. Although currently we don't have a relationship with Japanese National Cancer Center, we welcome you to reach out to us. And if there's any potential future collaboration, or if there's any project that you're interested in, working with us, we're open to discussion for sure.
Thank you, Wayne. Thank you, Shiping. And
I think I can add on a little bit. Okuda-san has a very close relationship, in fact, with the Japanese NCC. He's a very good friend of mine. I know that he's very well established in this field. So maybe in the Japanese market, we can have more dialogues and discussions with Okuda-san in the future.
Absolutely.
thank you, Wayne. Thank you, Shiping, and also Okuda-san for your question. I believe we have covered all the questions raised in the Q&A box and also the chat box. And there's no raised hand in the from the participants. I think we have overrun about like 15 minutes. May I ask if there's no more questions? We would like to conclude today's webinar. But this is not the end of the webinar. AMDL, together with Pillar Biosciences, would organize more webinars in the future to introduce our upcoming products and services. So please feel free to leave us your contact information via this QR code and stay tuned if you haven't done so. Today's seminar video will also be available at AMDL website very shortly. Feel free to revisit it and share it to your colleagues and your customers. I believe at the end of this webinar, all of us now have a very clear understanding on Pillar Biosciences cancer NGS products. The ONCO Reveal lung and colon cancer companion diagnostic test and the solid tumor panel are now available at AMDL. If you have any question about today's talks, Pillar Biosciences products, and AMDL service, feel free to contact us via this channel shown on the screen now. To conclude, I wish every one of you a very good day, and hopefully we will see you next time. Thank you very much.